So I'll be presenting a project that was part of the African Society for Bioinformatics and Computational Biology Codeathon in 2023. So the project's titled The Crisis of Proteomics Reproducibility, a Bioinformatics Perspective. So the study was inspired out of frustration and a bit of anger. So we originally wanted to do a comparative analysis of proteomics data for patients with thalassemia. But the authors gave very little information. We could not reproduce anything. Even after emailing them and them giving more information, it was still not enough. So we were very frustrated, and the codeathon is very short, so you have to move quickly. So we decided to then focus on reproducibility and trying to identify tools that are useful and what you need to reproduce these studies. And we kind of figured out that there's an overwhelming amount of tools, which you'll see later on, and you don't know which tools to use. There's very few tutorials on proteomics. The papers themselves, also the same tools are used the whole time, even though they're not very good. So there's limited information on appropriate tools, especially since there's no standard. So proteomics is the large-scale study of proteins, and it is very vital across many different disciplines, from health to food to agriculture. And compared to the genome and transcriptome, the proteome is very difficult to study since it's dynamic. And this is because the proteome can change over time. Different tissues in the body will have different proteins identified, and the proteome changes in response to stimuli. So despite this, it does provide valuable information that can sometimes be missed from genomic or transcriptomic data. But obviously, one issue is reproducibility, as mentioned. And this is largely due to inconsistent protocols, poor data reporting, and very complex software. So what exactly is reproducibility? There are a lot of terms that people use, and sometimes they're used interchangeably, but you can see the difference. I won't go through everything. But reproducibility is basically having the same data, using the same type of analysis, but different labs. So I want to do an analysis, and someone in America wants to do an analysis. Are they the same? Can we reproduce the results? Will we get the same results? So some of the challenges that come with reproducibility is, first off, the pre-processing. So before you even get to analysis, you have to pre-process your data, and there's no standard on how to do this, and studies also don't tell you how to do this. So you have to basically do quality control and fix your data before using it. And then there's obviously issues with transparency and reporting. Authors don't give enough information. There's no access to the raw data or code, and something called black box pipelines, which is common in proteomics, where you put in your data, you get a result, you have no idea what goes on in between. And then software diversity, as mentioned, there's a lot of tools, a lot of versions. Sometimes they're updated, sometimes they're not. They're incompatible with each other. Very frustrating. So there are solutions that are being done, especially in the past 2 decades. So things like Nextflow or workflow management systems, cloud integration, running the tools on the cloud. AI is becoming quite a big thing. Diane, for example, uses neural networks for peptide identification. More and more tools are becoming containerized, so that helps with version control. There are better reporting standards and more consistent data formats. And then obviously the community helps a lot, having GitHub, having forums, sharing and collaborating. So the aim and objectives of the study was then to assess the reproducibility basically of available proteomics tools. So this was done by basically getting all of the metadata from the BioTools registry, and then we did filtering and categorizing of proteomics tools, and then we scored these tools using a reproducibility scale that focused on FAIR principles. So for the methods, we used the BioTools API. We used a Python script that basically extracted all of the information listed below. BioTools is pretty cool. I think the majority of tools are available on BioTools. There are just some inconsistencies which we'll get to, but you can search basically keywords, and there is a website, so you don't need a script technically, but it's easier. So the only keyword we used was proteomics. So then we filtered the tools because there were 2,223, and I didn't want to go through each individual tool to kind of score them. So we kind of threw out all the tools that might have a poor reproducibility score. So we only kept open access tools. We threw out tools that were related to genomics and transcriptomics. We kept only command line workflows. So we threw out the GUIs, we threw out the web browsers, the web applications, that kind of stuff. And then we removed any tools that had terms that we weren't really interested in, so non-proteomics related terms. Then we searched or we checked the URL for each tool. If it produced an error, we threw the tool out. And then 605 tools remained. That's still too many. So we then looked at the citation count, which is how many times the publication was cited, and we removed any tools that had less than 50 publications. And that left us with 107. And then while going through the scoring, we found that tools still had missing information, inconsistent URLs, or the tool basically didn't exist, but the URL worked, but it led to nothing. So then the scoring system, as I said, it uses FAIR principles, so findable, accessible, interoperable, reusable. And there are 10 criteria. So for findable, was the tool archived, like in Zenodo? Is there DOI available for the tool? Was there version control, or is the tool deprecated? Is it well maintained? And regular reporting and updates. Then accessibility, can you look at the code? Can you download the tool easily? Is it open source or proprietary? And then how is the community engagement? Were there multiple forums? Is there a GitHub with where people are allowed to do pull requests, or is there a tutorial that people can look at and then an email to contact? So then interoperable workflow management systems, so NFCore, SnakeMake, Galaxy, and then does the tool take standardized data? So for example, proteomics data is very large, so it will come in like a WIF file, but no tools take WIF files, so then you have to convert it to MZML. So can you do that easily, or is it a hassle? And then for reusable, was the tool regularly tested? Was it only tested at publication? Is the tool containerized or partly containerized? How good is the documentation and manuals? Can you follow what's going on? Are there examples? Is there enough information? And then, can it be used in the cloud? Can it be used with HPC? Or is it just a Windows application that you have to download and use like that? So, because we threw out a lot of tools, it's not surprising that it looks pretty good with only 14 tools producing very poorly, with 31 tools having level 3, so good reproducibility. And as I said, that's because we threw out so many of the bad tools. Okay. And you can see that the tools that had good reproducibility, there's a wide range of what categories they fall into. So peptide and protein identification was high across all three, but for good reproducibility, that was a common category of tools. And then a lot of the tools, so COMET, MSGF+, peptide profit, all of that is part of something called the trans proteomics pipeline, which a lot of labs do use, and it's pretty good. A lot of it's containerized, it's very consistent, well maintained. So the reproducibility crisis is still around, and it probably will be for a while, because there's really a lack of standards and kind of what to follow if you're just starting out. But the community and having open source data and code really makes it easier to achieve reproducibility. And while reproducibility is important, a tool also has to be usable. A lot of tools are difficult to follow, difficult to understand, a lot of steps involved. It's not always well documented. So while we want reproducibility, we also just want to be able to use the tool and not struggle for months. And then just because something's reproducible doesn't mean it's popular. So something like Search GUI is kind of reproducible, but it's very annoying to run. It takes hours, it takes a lot of data or a lot of resources. So a lot of labs still use it because it's common and it's the norm, but that doesn't mean it's very good. And then tools are largely based on individual studies. So even with that, there's no consistency across the same types of studies. So it's hard to achieve reproducibility. And as mentioned, with usability, it's difficult to use a tool if there's no documentation, if there's no tutorial, if you can't ask questions. And then maintaining the tool is difficult. So someone will graduate, leave the lab, and then the tool just doesn't exist anymore, even though it was a really good tool. So with that, there were quite a few limitations. Using only one keyword limits you to only proteomics or tools that are annotated as proteomics. So maybe using more keywords would help. It would increase the amount of tools that you do get. Then in terms of the filtering, there's always going to be missing information, and that's not really something you can fix. And it's tricky because you might throw out tools that are really good, very reproducible, but they just have a few criteria that have missing data. And then some of the tools had non-functional URLs, but they're very good tools. It's just no one updated the BioTools registry. And that's also an issue, that needs to be maintained as well. And then with the citation count, it was very stringent. Less than 50 citations was thrown out. But older tools will have more citations because they are more well developed, they have been around longer. Whereas a tool like Diane, while it's new and really good, it will have fewer citations because I think it only came out like in the past 5 years. So then you can do something like gradient filtering, where tools that are newer, you only throw out if there's less than 10 citations, and if they're older, you throw out if there's 100 citations. And I'm running out of time. So basically, the study introduced a reproducibility scoring system, which is pretty helpful in deciding which tools you want to use, and if you want to create a tool, what criteria you can use to create it. And we basically kind of came to the conclusion that there needs to be standardization in proteomics. There needs to be more data sharing, code sharing, reporting, all of that, and more collaboration between labs as well, and between who develops the tool and who uses the tool. So thank you.
We've seen in audience and it's been certain things done and I don't know what the demonstrate about. But is there value add in approaching journals that really become the point at which articles are submitted? So is there a recommendation that can be made to journals as to what a paper needs to include? So we've seen this often in the audience space, that your paper needs to be accompanied by the accompanying some report will be accepted. You know that's
Yes, so there's a similar to transcriptomics or RNA-seq and genomics, there's a minimum information standard protocol, which tells you what needs to be included, but it's not really regulated by journals. There's a tool or an initiative called CodeCheck, which tries to check if the code actually works when publishing an article, and you can get like a little certificate saying your code works, and they're trying to kind of implement that in publishing or having articles accepted. So there are reporting standards, but there's no, it's not checked very well, which I think is common across all transcriptomics, genomics, and so on. Yeah.
So I have a couple of questions. What are and my question is regarding scoring of manual scoring or
Okay, I'll answer the second one first. So that's why I wanted to get rid of as many tools as possible, because there was a bit of manual scoring. It's difficult to check each thing individually because it's not reported very well. The data isn't there to automate it. So I went individually to choose the criteria that it matched, and then wrote a script to score across the table. So if it had this number of level 3, it would be level, like average out the scoring. For the first one, trying to streamline your pipeline as much as possible helps. Something like NF-Core really tries to make it easier and to make pipelines and bioinformatics a bit more usable. I really, with proteomics, there are so many tools that are just used, and a lot of them are GUIs because it's just easier, but it's difficult to then make that reproducible and to share it across labs. So it's difficult. I think we could learn something from transcriptomics and genomics, but it's also, proteomics is so much more complex, and it's so individually based on the study that it's difficult to have a standard, have something common, and yeah, well maintained. I don't know if that answers your question.
Thank you. But I'm just curious. I'd just like to know your idea or what you think about the tools that you rolled out. So I guess you have quite a number of evidence for the past. So do you think that for the publication when they use those tools, would you think that it's common to trust the results in us or otherwise?
I wouldn't say that you can't trust the results. This is very subjective, in my opinion. It's almost a suggestion of what you should do, but it's difficult to say that this is now what everyone should do, because there are so many tools that were filtered out that are used so much. So yeah, the scoring is, I want to say it's a suggestion, not a definite thing, but it's something that when you're wanting to use a tool, you can look at the scoring criteria and decide for yourself individually on that tool if it's good or not. Yeah, it's definitely journal specific.